Hello guys, um, welcome you all. I welcome you all to Bosons Botany. But before I start, I would like to say I'm extremely sorry for not uploading videos. I think for 1 or 2 months because I'm taking my exams, my final year exams. So I'm extremely busy with my studies. So I am not getting time for my channel. But I would like to thank all my sweet subscribers who are constantly there with me and supporting my channel, my work, my videos, and they're you know giving their nice comments. So I'm really thankful to them. Just stay with me and support me. Whenever I get time, I will upload videos, not to worry about that. So I'm starting with molecular biology today. I think I have not completed morphology in flowering plants. That I will do in between. But I think that molecular biology is a very important subject. It is not a topic or not a chapter, it is a subject in biology, and without this you cannot think anything else in biology. This is the fundamental subject we have in biology. And this is again a very difficult topic, I think, because you need to understand every bit of it, and without a proper teacher or a proper you know uh, guide, you cannot like understand this subject. So I'm really thankful to my professor who has given me extremely unique and very important notes on this subject. So it's a kind of gratitude I'm showing by uploading this video. So thank you, sir, for teaching me so well, and I'm extremely comfortable with this subject now, and I think that I'm you know well enough to carry forward this legacy of sharing knowledge or sharing the views on this subject. So without any further ado, I'm starting with molecular biology. So first I will take up a very basic topic, which is chromatin. So chromatin is basically this, as you can see I have drawn here. It is a thread like structure. It is a very messy, a very thready, or a meshwork type of meshwork network, you can say, inside the nucleus. So this is a chromatin, or you can say chromatin fiber or chromatin net, by just observing its appearance. This is nucleolus, and this is the nuclear membrane, and this is a nucleus. So chromatin is basically present inside the nucleus. So chromatin, nucleolus, nucleus, all are normal metabolic cells. Without them, no metabolism can take place. So they are very important and form integral parts in our metabolic system. Composition of chromatin. It is basically composed of two main ingredients, you can say: nucleic acid having DNA and RNA, proteins, histone and non-histone. These terms are quite familiar, I think, but I will take up them very differently, in a very different way, separately, in detail. So don't worry about that. DNA and RNA and proteins. The chromosome, or nucleic acids, as I prefer to write in short form, this is the abbreviation of nucleic acid, are not clearly visible but present as chromatin. So basically chromosome is present as chromatin fibers. So you cannot observe a chromosome, the actual structure of chromosome, because a chromosome is present as chromatin. So W. Fleming for the first time had introduced the term chromatin. Later on, Mischer studied chromatin in detail with nucleic acids and gave the term as nuclein. So these are some pioneer scientists who came and gave their contribution for discovering chromatin and various theories related with this. So for the first time, W. Fleming came and he introduced the term chromatin. Later on, Mischer, he gave his research works, and he made some important studies of chromatin along with nucleic acids and termed it as nuclein. Later on, Mischer isolated nuclein in reference to DNA from pus cells, thus reported the existence of nucleic acids in the form of DNA. So Mischer, on his further experiments, he had isolated nuclein from pus cells along with DNA, and from that he made a conclusion that okay, nuclein, which is basically chromatin now, as we know, nuclein has DNA. So he also isolated, purified, and crystallized these you know the contents that he had got from pus cells along with DNA, and discovered the presence of DNA. He also isolated, purified, and crystallized the nucleus and many other nucleoproteins. So during the course of his experiment, he also did all these processes or all these procedures in which he had isolated nucleus and also several nucleoproteins. Altman termed nucleic acid in place of nuclein. So Altman gave the actual term that we know today as nucleic acid in place of nuclein. So chromatin is composed of nucleoproteins. Nucleoproteins, if you break this term, nucleo means nucleic acid and proteins having histone and non-histone. So nucleo having DNA and RNA, proteins having histones and non-histone protein. Together they form nucleoproteins. So chromatin is composed of nucleoproteins. So nucleoprotein chemical composition. What nucleoprotein actually has? So there are several important chemical compositions like 36.5% of DNA, 9.6% of RNA, 37.5% of histones, and 10.4% of non-histones. So there is always a confusion whether DNA dominates the chemical composition in nucleoproteins or not, but basically histones are the dominating factors or dominating chemicals we have in nucleoprotein, because histones are nearly about 37.5%, but DNA 36.5%. So you need to remember the chemical compositions of nucleoprotein, because it's very important. No need to memorize the values of their percentages on and all, but just remember that DNA is not in its maximum amount, but it is histone protein that is present in its maximum amount. Recently, calcium ions are supposed to be in nucleoproteins. So it is a recent discovery that is made where scientists have introduced or they have found the presence of calcium ions in nucleoproteins. Histones, as I have already said, with maximum percentage are mostly basic, having monomers or monomeric units of basic amino acids like lysine and arginine. These are two basic amino acids which are present in histones. Therefore, histones are basic in nature. During cell division, the chromatin net converts itself into chromosomes. So, if you observe a nucleus during its division or during cell division, you can actually observe fine structure of chromosomes, because at that time the chromatin nets undergo a modification to change into chromosomes. So I have drawn this schematic representation here, where this is a nucleus, this is chromatin net, this is nucleolus, and during cell division it is converting into chromosome. So this is a chromosome, and the structures that are present here are two chromatids down and two chromatids up, one centromere, which is also known as the primary constriction, which is towards the center, and there are two chromonimata. These are thread-like structures, as you can see I have drawn here. So just to wind up this concept, one chromatid has half chromosome and one chromonimata has 1/4 of chromosome. So as you can see here, one chromatid will have half of its chromosomal contribution, or half chromosome, and one chromonimata will have 1/4 of the chromosome. I think this is clear. Later on, A. Ollins and D. Ollins had reported the existence of beaded structures along chromonimata, along the entire length of chromonimata, and later on Woodcock termed these beaded structures as nucleosomes, also known as new bodies. So now I will take up nucleosomes. So nucleosome, as I have already told you, nucleosome or nucleosomes, whatever you want to say, these are beaded structures present on chromonimata or along the length of chromonimata. So this is a thread of chromonimata, and there are nucleosomes present as beaded structures known as nucleosomes or new bodies. If you further enlarge the view of this whole structure here, then you can see this is a DNA which is wrapped by 3/4 turns. So this is the number of turns a DNA forms around the histone proteins, and there are 14 base pairs present in this 3/4 turns of DNA. And here are 4 circular structures, these all together known as the Robin core of 8 histone proteins. So this is a terminology that is used for these structures here. Basically there are 8 such structures altogether known as Robin core of 8 histone proteins. So Harvind Khorana and Thomas had studied them in reference with composition of DNA and histones. So Harvind Khorana and Thomas, they actually did their studies to reveal the chemical composition of nucleosome along with DNA and histone proteins. So new body or nucleosome basically has DNA showing 3/4 turns plus 3 H3 2 and H4 2. So histone 3 and histone 4 present 2 each. So here as I have written 2 in the subscript here. So histone 3 and histone 4, 2 of this and 2 of this, together forming 4 tetramer core. So 2 H3 histone protein and 2 H4 histone protein forming tetramer core plus H2A, H2B. So these form oligomer core. So tetramer core and oligomer core, these two altogether form the Robin core of 8 histone proteins. So again, new body has DNA which is showing 3/4 turns or wrapping around the Robin core of 8 histone proteins for 3/4 turns. Also has tetramer core, oligomer core. The tetramer core is present in the center, whereas oligomer core is present for giving spacing needed for stability. So tetramer core is present in the center and oligomer core gives spacing for stability. Audet et al means his co-workers, as I have already defined this term in my previous videos, so you must be knowing this. Audet, along with his co-workers, found and studied the biochemical nature of nucleosome. Later on, Klug studied it along with its crystallographic structure. So these are some important points regarding nucleosome, and I have drawn an enlarged view of nucleosome here. As you can see, this is self-explanatory, but still I will define its enlarged view. So this is H2A histone, this is H4 histone, this is H2B histone, this is H2A histone, this is H3 histone, this is H2B histone, this is H4 histone. These are H1 linkers, and this thread that is composed of DNA, it is known as internucleosomal DNA or spacer DNA. So there are some few points left regarding nucleosome that I will complete right now. So remaining points or last few points about nucleosomes are 8 octameric core, which is composed of histone proteins present in the center, and this is wrapped by DNA coiling, which is 1 3/4 if you remember. So the 8 octameric core, which is composed of histones, is wrapped by DNA coiling. The H1 histone protein separates the two adjacent new bodies or nucleosomes from each other. So the linker H1 protein, if you remember the enlarged view of nucleosome, this protein basically separates the two nucleosomes from each other. Woodcock also proposed the perimeter of one nucleosome, or the circumference of one nucleosome, which is basically 11 into 11 into 5.7 nanometer in size. So this is a very unique point that I have given you. Each new body is a flat cylindrical structure, also known as quasi-cylindrical. Average diameter is approximately 10 nanometer. When new body is further coiled with complexity, then it is known as solenoid model of nucleosome. I think this is a familiar term, solenoid model. I am not discussing it here. So due to further complex coiling shown by nucleosomes, a solenoid model is obtained. So this is all about nucleosome. In my next video, I will take up nucleic acid having DNA and RNA. Very, very important. I don't know when I will get time, and I have no idea about my second video, but I hope to make a video on nucleic acid very soon. So if you have any doubts, any problem, any queries, please comment below this video, or you can also write on my any other video comments if you have watched. So just give your questions, give your queries, because I regularly check my email and I find several queries and also good comments. I'm really happy for all these things. Just give your comments and give your thoughts, your questions, and I'm definitely there to help you guys. So just support my videos, stay tuned with me. Thank you.